Hello everyone and welcome to this roundtable. So as just stated, we'll be talking about leveraging AI or GenAI, we'll see, in genomics and beyond. And I want to welcome, warm welcome to those three gentlemen who are as different as they are complementary. So we have Ramzi. I will let you introduce yourself and why you're here, because you had several lives. So.
Sure. Ramzi Temanni, I'm leading the AI for Systems Biology group. It's part of data science and digital health in the R&D organization within J&J. I'm also in charge of public-private partnership in France, leading the AI in Genomics initiative. Previously, I was also leading the bioinformatics core in charge of the Qatar Genome program. I've worked also in the US on dual RNA-seq and host pathogen interaction, spent some time in startups working on next generation analysis pipeline. So yeah, I've been on the multi-omics for some time, working on several projects.
Thank you. So yeah, you hear quite a lot to share with using genomics. And then we have David, who's our expert as the CEO of Whitelab Genomics, and I will let you briefly introduce yourself.
Hi everyone, thank you. Happy to be here. I'm David Bourgo, CEO, co-founder of Whitelab Genomics. I'm a healthcare veteran with 25 years of experience in the field of life science and created Whitelab Genomics. We're an AI company specialized for the development of advanced therapies, in particular genomic medicine, therapies using DNA or RNA. So we're doing the in silico simulations that we provide to our clients, pharmaceutical companies.
Thank you very much. And last but not least, we have Amaury Martin, who has several hats, but today it's as the president of Seqoia that you're here, right?
Yeah, exactly. So, hi everyone, I am Amaury. I am the deputy director of Institut Curie. I am in charge of data, tech transfer, translational program, and also administrator in French, but president in English of Seqoia, which is one of the two platform of the France Medicine Genomic Plan 2025.
Thank you very much. So I think genomic is a staple when talking about AI for health, as it's definitely one of the first illustration and of using big data. So starting with millions of base pairs, you should start sequencing a genome. And thanks to the technologies that has very much advanced lately, thousands of individuals, you know, if you time that with all those base pairs. And on top of that, now we also have other kind of omics giving more information about this genome, those genomes. So it's getting more and more complex, but it's a perfect example of big data, using big data. I was hoping maybe, Ramzi, with your past experience, if you could give us maybe a landscape of, you know, what is available today, what is at hand, what is maybe it's still hard to reach, if you could give us a landscape.
Sure. I think I'll start first by saying that back in time, the cost of sequencing a genome was exorbitant. So we're talking about 15,000 euro back in 2009, around that range. And today, the cost is really going down drastically, which make it easy to generate more data. There's been also a big improvement in data quality, the long length of the reads, and also the capability to kind of process the data, I would say more on the bioinformatics stand of view. I remember like back in time, it took like few hours, maybe 6 to, let's say, 18 hours to do the mapping and the variant calling. Nowadays, with new technology, you could have that in just half an hour. And not only the quality of the data or the sequencing is getting better, the tool also improved a lot. A lot of databases that help to get a better annotation are available. There's one of the aspects that people were considering, as we call the junk DNA, that turns out is not junk. And I think it's one of the area where GenAI helped, for instance, to predict the non-coding structure of those regions. The other things also that makes you know, the field advancing is also the advent of the RNA transcriptome and also single cell analysis. And those also, there's a fascinating technology, emerging, even in proteomics, where now we have sequencer that can perform like single molecule protein sequencing. And I think with all these advances comes challenges. We want to get the data faster. We can be able to process the data. But ultimately, those still things that are only accessible to bioinformaticians who have like the right competency. If we want to make them accessible to a more general scientist, I think GenAI here can play an important role in that area. Like David, if you want to add anything.
I mean, I was gonna say the same thing exactly. Well. So I was like, what am I gonna say next?
No problem.
Raphaelle, I let you.
No worries. So yeah, it's definitely in a place where we can actually really use those data thanks to those technologies today. And sorry, and Amaury, if you could give us a practical example with Seqoia and what you're doing at Seqoia.
So yeah, just some words about Seqoia. So in France, we had a chance to be involved in a plan, national plan called France Médecine Génomique 2025. And the objective was and is to put genomics into the clinics. So it's not easy, but I mean, that's a long time we were doing sequence for cancer or rare disease. But it was not included within the treatment plan. So that was the objective of the plan. And so it was launched 6 years ago, really. And it's doing both rare disease and cancer. Around, we have 70 indication, what we called. So people clearly treated within a hospital that can enter this process. And then before getting a diagnostic, you have the data, genomics data, to help the physicians to put the diagnostic and to discuss what should be the best treatment plan for the patient. So that's really a specificity of this plan. And to my knowledge, we are on the forefront in France of this, because no other countries have tried to do that. So what we can learn about 6 years after, that first, it is possible to do that. It was quite hard to set up all the processes to mobilize the different clinicians, the different biologists. So 1,300 biologists are involved only in Seqoia platform to be able to detect the patients and then to send the samples to the platform. On now data, it's 3,500 persons that were diagnosed with cancer with clinical reports, not only prescription, but really diagnostic. And it's 12,000 persons with rare disease. So it works. On the one question you can ask is the delay. So that really depends on the indication, but it's possible to have a full sequence analyzed within 2 weeks up to 8 weeks. But that's not bad. I mean, that's compatible with a treatment plan. On rare disease, it's a bit different.
And so you achieved a lot in quite a short time. I mean, we all know that those things, you can have so much challenges that it's very impressive. Well done. And so now that you have all this data, what would miss, according to you, what is missing today to really leverage what we could get from those data?
Yeah, go beyond all of that. Well, obviously, and I'm not going to discuss that because I can get a bit upset, is clearly GDPR, the way we can share data. But I will let my colleagues say something on that.
Complain about
because we have the data now, it has to be used. So that's the point. The second one, if you think a bit ahead of that, so I mentioned for the cancer, now the flow, let's say that, we estimate that it's possible in France to be able to sequence, let's say, 4,000 patients per year, which is not bad. But if you look a little bit, what is the reality? We have 150,000 deaths by cancer every year. So if we want to let's say, have 100,000 person diagnosed with sequence, I mean, this is a huge step that we have to be able to make it. So that's one of the major challenge that we have to overcome. This is the first one. The second one, and I will let also my colleague elaborate on that, is the connection with clinical trial, because it's a good point to say, okay, we have this mutation, but then, I mean, what should be the best treatment plan? What should be the best therapeutic option? And here we have it's complicated to connect things. And the last one, and I think not the least, is that we have this amount of data to be analyzed. So some of them are easy. Do you have the mutation, yes or no? But some of them are not. That could be a strong chromosome rearrangement, mutation. And then my question to the experts is, I mean, can we rely on AA? Can we use new technology to speed up things? Because that is clearly now the point. It's not the cost that was mentioned before. It's not the delays of sequencing. It's really the analysis of the genomes and the possibility to put that in the treatment plan compatible with the disease itself.
Yeah, I think we are far from Mendel's peas and black and white, and whether we will have blue eye or brown-eyed kids. Definitely, and this is hence the beyond of this title. It's that it's not just, and also Ramzi mentioned the junk DNA. On top of that, you have all the epigenetic and obviously the transcriptomic, and combining all this information is definitely in the best way. You can do it in parallel, but then how do you make all those layers crosstalk and give you far more powerful insight? And so yeah, I'm turning to you, David, if you could give us some example with what you're doing.
Yeah, very good. And just to build up on what's been said before, it's true we are a new phase in the field of how AI can help in healthcare in developing new drugs, because there is. You always want more data, but there is a lot of data. They're more qualified. There are the tools to analyze the data now. There are the computing power that's here, that allows to go to half an hour for analysis, for complex analysis. And so, to answer your question, is because there is so much diversity also in the data, you need to be focused. So the way at least we do it at Whitelab Genomics is we're going to look at a specific disease, a specific cell, and then we're going to analyze the multi-omic data that corresponds to a specific cell type. If we want to treat a patient, for example, if we want to treat AMD, age macular disease, eye disease, we're going to analyze the specific cell type that corresponds to the degeneration in the eye, and then build a therapeutic strategy in silico, in the machine, so with a lot of computing power, a lot of AI models, whether you use protein language models or generative AI to generate new sequences, and then they will be tested in the lab. So this is how you speed up the treatment design, because you're going to do a lot more in the machine than in the lab. And we all know the lab costs a lot of money. I mean, the GPU usage also costs a lot of money, but it's less capex in the end. So I think this is where you accelerate the way treatments are developed, and I think it's a very exciting time to be, because we can look beyond. There is, again, computing power, expertise, a lot of AI tools developed, also by, AlphaFold, type of tools, and Google DeepMind, and so forth, and many other initiatives. So I think it's a great way to a great place where to be right now, to accelerate and do what we can do best to bring more treatment to patients, whether it's oncology or rare diseases.
Thank you. So speeding up the design of the drugs. Thanks.
So maybe I'll bring back the slide when I present AI in genomics. We have 4 pillars. So the first one is the data, the second is the algorithm, the third is the platform, and the most important is the talent. So if we analyze one of those, and there's no one fit all, so we see that, for instance, in terms of platform, there's huge advance. If we talk about computing, for instance, beyond the NVIDIA, we heard about Cerebra, we have about Grok. So this new LPU might speed up whatever is happening with generative AI. So that it checks a box maybe to reduce the cost, get access to new technology. If we talk about the data, we have 2 paradigms. either a well-targeted cohort that is of interest to a specific question, and then you're trying to create multimodal data on that cohort to better understand a specific disease. And on the other side of the spectrum, you have the population analysis where you have a lot of data. The challenge there, again, genomics maybe is the easiest because, again, genomics data doesn't change that often. So it's a snapshot, then that doesn't move much. When we get to the transcriptomic, you're taking a picture in a video. So there's also a lot of nuances. Are we taking the right picture at the right time for the right population? So it might be there's a lot of work to be there to make sure that we get access, maybe not a lot of data, but a good data that is really related to what we're trying to do. When it comes to the algorithmic, I think there's a lot of work because, again, like the AI was there, there's a lot of application of AI. GenAI is coming, it comes with a lot of advantages. And also it comes with challenges. Hallucination is getting better. However, I think the challenge in this area is how would you validate the outcome? Because basically if you generate text, you would know whether it's right, either it's like a general text or it's like a specific text related to a field. You could bring an expert and tell whether it's wrong or right. Now, if you take it to genomics, it might be that the way to validate the work will be exorbitant or hard to generalize if you do it in few examples. And then the last point, which is the talent, and that's where we see that today's problems are complex. And the most important thing is to bring around the table multidisciplinary team that could think out of the box to work together to make sure that you have the right results.
And that can actually talk together also.
Absolutely.
Yeah.
So you need this dual lingual or bilingual talent that could know the biology and also the data science. And I'm lucky to have those in my team. So,
thank you very much. We have the technology. Let's say we have the talents. We have some data. And so and the platform. That would be your fourth pillar. It seems like we have everything. Okay, so what is it you think that is kind of blocking us that could be maybe blown up very soon? Maybe, or maybe what would be your wish? You know, what you would like to really overcome. If there was one wish, maybe I'll start.
Oh, my wish is the way we. In France, I'm going to be French-centric.
And remember, the ministry wants the summary
don't
after the day.
The way GDPR is applied in France is, for us, really complicated. I mean, the need of having systematically the patient saying, okay, yes, I am agreed to share this data. So the idea is great. But now we spend more time on tracking the different agreements of the patient for one study, for another study, for the third one, etc., etc., etc. And at the end, it's really blocking the way of how can we take this amount of data to really generate a new concept, find things. So it's a shame because it was done to protect patient, protect privacy, and at the end, it's slowing up things. And so that my wish would be that we say, okay, we can exploit the data, and if you don't want that your data to be exploited, okay, say it. It's but just do it on the reverse. So that would be my wish if you want to speed up things.
So you don't see it, but there is a sign just behind it: free data, free data. And I think that's a shared thought for everyone. Yeah, David is preparing this sign. David, what would be maybe you're allowed to have the same wish?
Yes, it's very similar, and it is of course around like freeing data or getting access to more data. One example maybe easy to understand is that if you look at diseases, it's like we have a lot of data available in genomics or, transcriptomics or proteomics, like our public repositories, but a lot of like standardized, healthy tissue, healthy cells. And of course, you can really easily understand that if you want to target non-healthy cells, they have a different shape, they have a different structure. And therefore, if you don't have this information, it's harder to access it, right, and to create a drug that's going to be efficient. I think for some rare diseases, for example, you have very little data for on patient data, and sometimes it's really heartbreaking to see you cannot really create this drug because you don't have enough data to create the drug. And we see there will be a Téléthon in a couple of weeks. You see like those miracle treatments, you know, that restore eyesight, that restore muscle function, and we can do more of those, right? But we need more like patient data and disease state cell type.
We should have brought some patient experts to the round table next time, maybe. Ramzi, what would be your wish?
So I'll give an analogy. So I talk about the ingredient. So imagine you're in the kitchen, you have all the ingredient to cook, but then you have a sign on the fridge, another sign on the stove, and with a lot of restriction. And again, like you have a big manual where you have to read everything and you sign off and you do things. So it's exactly the way I characterize the situation. So having the ingredient is important. I think it's the whole ecosystem that is outside of R&D that is connected to the question that is the challenge. And again, it comes back to the ability to understand the different stakeholder and them understand the research, trying to find the right balance between privacy and utility. And most of all, you know, like having someone behind to trying to get all the actors of the ecosystem together in kind of synergetic way to make things advance. But again, because the problems today, although we have a lot of data, there's a lot of intuitive method and we have talent, they are challenging. They're not easy and might require really pluridisciplinary team, collaboration between public and private to make sure that they have a win-win situation that could help everyone to advance in support of the patients.
Okay. Sorry?
I can make
Sure.
I think one more wish.
Go ahead.
I wish that
Almost
Davis
Christmas.
gets more fund, because I think that the key on all of what we're saying is that if there is more investment in tech bio companies, that's we have all the technology, we have the talents, we can unlock a lot of things, but at the end, we just need money to do those things. It's basic.
Est-ce que vous m'entendez?
It's not that. I mean, we can spend a lot of money on things that are not really useful for the.
Ce qui va se passer, c'est que là,
us,
comme c'est
for
en
the
train
people,
d'évoluer,
but if we can move a
je ne peux
little
pas
bit money
je peux pas
on
attendre
that,
votre échange
that could be
avant,
the,
parce que le problème, c'est que ce que vous allez dire va
I
s'entendre
think
sur
we
scène.
should be in the next,
Donc là, je vais vous
audits
laisser
of,
là à regarder ce qui se passe sur
Thank you.
scène jusqu'à votre intervention. La seule chose que je vous
Well, it's
demanderai de
actually
faire,
pretty good
c'est
news.
qu'il
If
faudra
I
penser
hear
à
you,
désactiver votre micro,
we have everything.
à le réactiver au moment de
It's
votre prise de
just a
parole, parce que pour
matter
l'instant, je le vois éteint. C'est bon? Merci
of
beaucoup.
will. So here is the message for the ministry for the end of the day. I hope it will be in the report. More money and more clever regulation, let's say. Maybe try to see it the other way around.
I might finish in one note. We were talking about the different modality, but I think the challenging part is on the multimodality,
Activez votre
trying to
micro
get
dès maintenant,
everything
comme ça
together.
moi je serai sûr.
Okay,
que
so
vous
let's be
pouvez-
honest.
And that's
Merci beaucoup.
basically
Let's be
the
honest, we
next
do also still have a bit of technical challenges, right?
Well, there's small challenges everywhere, but I think if you tell me what will be the next big jump is going to be the multimodality, and trying to have in mind all the small challenges on each modality in kind of a system biology thinking way, then probably the problem will arise and needs to think out the box to solve them.
Definitely. Well, thank you very much. Still have plenty of questions and things to address, but I think we're going to run out of time. So we'll stop there. But thank you very much. That was a very interesting conversation.
Thank you.
Thank you very
Bye-bye.
much.